Okay, I guess we're live now. Yes, I checked it on the YouTube site also. So let me then get started quickly. All right, welcome everyone. Good morning. Today we're going to present two of our recent works. I'll first start with RawHash, a tool for mapping raw nanopore signals to reference genomes. And the second work is going to be presented by Banu, a work called TargetCall to quickly filter out uses nanopore raw signals without fully base calling them, let's say. So, but mainly in both of these lectures, we're going to be focusing on the nanopore raw signals and nanopore sequencing. So that's why I'll also give very quick background on the nanopore sequencing during the first lecture. So with that, let me start. All right, yeah, as I said, I'll present RawHash, enabling fast and accurate real-time analysis of raw nanopore signals for large genomes. You can find the preprint of our work using this QR code, and you can also find the source code thing using this QR code as well, if you're interested in. So let me quickly start with the executive summary. So if we are using nanopore sequencing, there are certain advantages of nanopore sequencing. For example, we can perform real-time genome analysis as the raw nanopore signal data is being provided. And there are some tools doing such an analysis, but these tools are essentially, they are either inaccurate or inefficient especially for large genomes. And these issues causing serious barriers in fully exploiting the opportunities in real-time genome analysis. So our goal in this work is to enable efficient and accurate analysis, especially for large genomes, while the raw sequencing data is generated in real time. So RawHash has mainly two steps, and additionally, it proposes one novel technique to reduce the sequencing time and cost. So in the first step, the RawHash encodes the raw sequencing data into hash values so that we can accurately and efficiently identify similarities by simply matching their hash values. And after matching the signals to each other, it can also make real-time decisions that can stop the sequencing a DNA molecule without fully sequencing it, which is significantly useful for utilizing the flow cells or the nanopores that are usually used in this nanopore sequencing fully so that we can avoid sequencing useless reads. So we also propose in RawHash a novel technique called sequence until, that can accurately and dynamically stop the entire sequencing. So it's not stopping the single DNA sequencing of a single DNA molecule, but this can stop the entire sequencing of all DNA molecules at once. So RawHash provides up to 2x more accurate mapping results. It provides 25.8 and 3.4x better average throughput compared to the state-of-art works Uncalled and SIGMAP, respectively. And the sequence until technique enables reducing the sequencing and time and cost significantly. So let me briefly go over the very brief background on genome analysis, which we also covered in the previous lectures. So I'm going to relatively quickly go over this slide. In genome sequencing, by sequencing a DNA molecule, such a mechanism, it enables us to determine the order of the DNA sequences in an organism's genome. And this is essentially useful for many applications such as precision medicine, outbreak tracing, and understanding of evolution, etc. So as I said, we're going to be mainly focusing on the nanopore sequencing in today's lectures. And nanopore sequencing is a widely used sequencing technology. It has some novel features, which we're going to cover them. So nanopore sequencing can sequence large fragments of DNA from 10 kilobases to a few million bases. It has a relatively high throughput. It is a relatively low-cost sequencing technique, and it also provides some unique features. So let me quickly go over the traditional steps that we usually follow when doing genome analysis with nanopore sequencing. So in nanopore sequencing, we have a few parts that play an important role to sequencing the DNA molecule. So in this picture, here the middle part is usually known as the membrane, where we have like a steady current flow on this membrane. And you can sort of assume that the top part of this membrane is charged by is negatively charged in terms of the ionic charges over here, and the bottom part of this membrane is usually positively charged. And since the DNA is also RNA is negatively charged, so what happens is that physically these nanopores usually flow through these tiny channels that we call nanopores. They flow through them towards the positively charged sites. So as they flow through, they're going to disrupt the ionic current flow that was charged on this membrane. So that disruption of the current flow is then recorded as the electrical signals. But the idea is that whenever a new base is being, let's say, inserted to these nanopores, they're going to create a nicely identifiable, let's say, or recognizable charge disruption, such that we can identify the particular base that went into this particular pore at a time by simply analyzing these raw signals. So how do we usually analyze these raw signals? So by the way, these signals are called raw signals because they are usually in the form of the electrical signals. And what we usually do is that we translate those signals to the bases using a step called base calling. And these base calling tools are usually deep neural, they use deep neural networks to, when translating these signals to the bases. So they usually get like certain chunks of the signal, and then they converted them to bases. So after essentially translating all the signals to their bases, which we call them also reads, as you may remember from the previous lectures, what we usually do is that we want to analyze those reads, usually by mapping those reads to a reference genome. So as you may know, some reads may map to a reference genome, or some of them won't map, etc. But we can use this read mapping information to do some further analysis, which can lead us to enable some scientific discoveries by doing these further genome analysis. So this is a traditional step in the genome analysis pipeline. But if you remember, I also mentioned this real-time generation of the sequencing data. So we can use a unique feature of nanopore sequencing to utilize this real-time data generation from nanopore sequences. So remember, this part is negatively charged and this part is positively charged, and the DNA flows through these pores because the DNA is also mainly negatively charged. So these are essentially the physical, the electromagnetic forces happening during the nanopore sequencing. And while the DNA molecules go through these pores, they're going to generate these raw signals in real time as they are being sampled at a certain rate. So as they are being generated at a certain throughput based on a nanopore sequencer that we're using, then the goal is to be able to analyze that data that is being generated in real time. And to achieve this, of course, the main challenge is to match the throughput of the nanopore sequencers so that we can analyze it by being not too slow, let's say, in our analysis. Then what we can do is that we can make some decisions based on our real-time analysis. So these decisions are usually known as adaptive sampling, or you may also have heard read until, run until mechanisms. But what they essentially do is that they can let us decide whether we want to keep sequencing a particular read based on our analysis that we've done so far, based on the raw data that is being generated so far, let's say. Because let's say a read may not be mapping to a particular reference genome. If it is not mapping to a particular genome, then you may not want to keep sequencing it. For example, if you are doing some sort of contamination analysis and to check whether your sample is contaminated by a certain, let's say, species. And if the read is not mapping to the reference genome of that particular species, you may just very well stop sequencing it because you're really interested in seeing whether you have that particular genome in your sample or not. So why waste time and also money keep sequencing that user's read, right? So if you can make that decision, what we can do is that we can tell the Nanopore to eject that particular read from a Nanopore so that we will stop sequencing. So how does that happen? It happens mainly by reversing the polarity of the electrical field over here. If basically the voltage is applied at this position, you can simply apply the voltage over here so that the polarity is reversed, such that now this part is going to be positively charged, so that the DNA won't move towards here, rather it will be basically will move in a reverse direction so that it will be ejected from a Nanopore without sequencing it further. So this is again known as adaptive sampling, and it is essentially useful to reduce the sequencing time and cost by utilizing some real-time analysis tools. So then let's look at the main objectives in real-time genome analysis. So what are those objectives? So we want to do fast analysis that can match the throughput of the sequencer, right? Because if your analysis is slow, meaning if you are falling behind the speed that the Nanopore sequencer is generating the data, then this means that you're not really doing a real-time analysis, right? You're doing analysis for particular data that was generated in the past, and then if you make a decision based on the past data, then maybe your decision is just particularly too late. So the second objective is to do a fast decision to reduce the sequencing time and cost with effective use of adaptive sampling. So this means that we don't want to sequence the DNA for a very long time. Rather, we want to make the decision as quickly as possible by sequencing as few bases as possible. So the third part is to do accurate analysis from noisy raw signal data. We don't want to basically make incorrect decisions. And the fourth objective is to do all of these decisions using a low power mechanism to essentially enable portable sequencing and better scalability. So then let's look at some existing solutions for real-time analysis that's utilizing the adaptive sampling in Nanopore sequencing. So the first solution is using deep neural networks to base call and map reads as they are being generated. So how it happens is that as we generate the raw signal data in the real-time analysis step, what we can do is we can base call the data that we generated so far. So what will happen is that then we're going to be partially sequencing the raw signal, and then we can use that partial sequence data to do, let's say, further analysis, for example, read mapping, to decide whether that particular portion of the data can map to a reference genome. And then we can do a decision, right? The adaptive sampling decision if you want to keep sequencing. So in that case, let's say we decided we want to do, we want to keep sequencing the DNA, so we sequenced and then generated the second chunk of the Nanopore signal, and then we base called it again, and then did read mapping. And then now, let's say we decided that based on our analysis so far, we decided that we don't want to keep sequencing the DNA. So what we can do is that we can tell the Nanopore that it should stop sequencing. So it won't actually sequence the next part of signal, meaning it will save us from sequencing time and cost, hopefully. But the main issue with these type of works is that they are mainly costly and energy hungry because essentially they use deep neural networks or machine learning, costly machine learning approaches, let's say, to base call and map reads. So then let's look at the second solution, the second type of solution. In the second type of solution, we can map the signals without base calling them. So again, like we're generating our signal, then what we can do is that we can directly map the signal without base calling it. There are some certain techniques that enables that essentially. And again, after mapping the signal, we can ask the same question: do we want to keep sequencing it? Maybe yes, so let's keep sequencing it, and then analyze it again by retrieving further data, and then maybe we want to stop sequencing it. And similarly, we can utilize adaptive sampling to stop sequencing in real time. So these approaches are usually low throughput, or they are inaccurate because they are mainly dealing with the raw signal that has certain issues that I'll be covering in some of my next slides. So given these problems, let's look at our goal then. So our goal is to essentially realize all the real-time analysis challenges that I mentioned in my previous slides, which are: we want to do fast analysis that can scale to large genomes. We want to make fast decisions for adaptive sampling to reduce sequencing time and cost. We want to do accurate analysis, especially for large genomes, and we want to do all of these analyses using low power techniques or efficient techniques, let's say, so that our analysis can be used with portable devices. So to this end, we propose RawHash. The RawHash is the first mechanism that can efficiently and accurately map raw signals to large reference genome using an efficient hash-based search. We also propose a technique called sequence until. This is a novel mechanism that can dynamically decide if further sequencing of reads is unnecessary to stop the entire sequencing for certain genome analysis applications. So with that, I'll go over the mechanism steps in RawHash, but I'll quickly check if there are any questions on YouTube for me to answer. I'm quickly checking. I'm seeing no questions, so I'll simply move on. So then let me go over the steps we have in RawHash. So this figure shows the overview steps in RawHash, and RawHash mainly has two steps. The first step is indexing, which is an offline step, meaning we don't do this step in real time. So we could, like theoretically, we could also do it, but usually when mapping the reads to reference genome, usually there is little need to do it in real time. So we can do the indexing first at a certain time before sequencing the reads, and then utilize that information that we generated in the mapping step. So the second step is the mapping step. We map the Nanopore raw signals to the reference genome using the indexing information that we generated previously. And this step is an online step. It happens in real time. So indexing step, it converts the reference genome to its synthetic version of the signals. So it essentially converts the sequence to signals. And then by using the synthetic signal information, it performs certain steps that we call event generation, and then quantization. Then by using the values that we generate after quantization, it generates a hash value for particular regions of the signal. So those hash values are then going to be useful for us because we're going to be generating hash values also from Nanopore raw signals, and we want to match these hash values. So in order to efficiently match these hash values, we generate the hash values that we generate from the reference genomes, and we store them in the hash tables. So along with the position information where they appear in the reference genome. So this step is usually known as the indexing step, and this is also a common step, not only in real-time raw signal mapping, but also when mapping the actual base called sequences to reference genomes. So in the second step, we map the raw signals. What we do is that we again identify particular regions of interest from these raw signals, and then we follow like similar quantization steps and the hashing steps, such that we generate comparable hash values that can essentially be compared to the hash values that we generated from the reference genome signals. And then now we use the hash tables to query the hash tables that we generated to see if if we have the same hash value in the reference genome. If we can find the same hash value, then we find the matching regions between the Nanopore raw signal and the reference genome. And then if you have sufficient number of matching regions, then we can do some chaining that essentially chains the collinear matches of these hash values, hash matches, let's say, and then we map if there is a sufficient amount of matches, or if we can chain the hash value matches, let's say. So this is how we do mapping, mapping of Nanopore raw signals to a reference genome. So let me then quickly cover how we convert the sequences to signals. So our goal when converting these sequences to signals is to enable analysis between reference and lead signals without base calling. We essentially want to compare the signals to each other, not convert the signals to bases and then compare them in the sequence domain, let's say, because that sequence conversion needs to happen online if we're converting the signals to sequences, and it can be costly to do so. So we really want to offload the costly part to the offline step. And how we do this? We essentially identify the corresponding signal value of all overlapping fixed length subsequences that we call k-mers. So we extract all these k-mers from a reference genome. So let's say these are 6 mers in this example. And we usually have this lookup table that is previously generated, where we can check the table to see what is the expected, let's say, the signal value when if I were to sequence that particular 6-mer sequence. This is because it, of course, depends on the nanopore chemistry or the nanopore technology that we're using, but this is essentially because when you're sequencing a DNA molecule, we usually have a certain amount of DNA bases within a nanopore. And in some cases, like we usually have 6 bases. It again depends on the chemistry. And we can then map basically what sort of current signal that we may be reading when we have that particular 6 bases in the nanopore. So if we can sample or if we can estimate what sort of signal values that we could read from this particular 6-mer, that we can actually map these 6-mers to 6-mers to their expected signal values. So we're going to call them event values because theoretically each event corresponds to a particular k-mer. So after essentially converting these k-mers to their event values, then we normalize them by using z-score normalization calculations. This basically essentially enables us to for the later steps when matching the raw signals to the synthetic signals, to the expected event values of the reference genome, because we may be generating raw signals at different, let's say, scales, different offset values, although the closeness of the signal values can be similar to each other, as we observe also here when converting the sequences to the k-mers. So this was the step to convert the reference genome to sequences to their expected signal values. So now I'm going to cover how we identify the events from nanopore raw signals. So remember, those events are going to correspond to a particular k-mer because what we usually assume is that there is a certain amount of bases located in a nanopore, which we also refer as k-mers. And those k-mers are usually 6-mer, 9-mer, depending on the chemistry that we're using. So we call these then events to reiterate myself. So the goal in the read signal processing step is to identify the regions of signals corresponding to k-mers in a read. To achieve this, we perform a statistical test to identify the abrupt changes in the signal. So remember, when there is a new base coming into a pore, what we assume is that the current reading is going to be disrupted, such that it can it can be informative for us to determine what sort of new base coming in to the nanopore, creating a new chimer located inside the nanopore. So by identifying these abrupt changes, we can then identify each chimer located in the nanopore, doing these statistical tests. So doing the statistical test, usually known as the segmentation. So those segmentation techniques will tell us at which positions those peaks or the abrupt positions are appearing, such that we can then identify, okay, the signals between any two segmentation points correspond to a single chimer or a single event. Then what we do is that after identifying the events, we calculate the means of the signals that are located between two segmentation points, so that we can generate the value of an event from the average values of these signals. So after calculating the means, we then similarly normalize these values so that we can now basically compare these values to the normalized values that we generated when converting the reference genomes to their synthetic signals. So there is one important observation here that I want to mention, which is going to be important in the later steps. Nanopore sequencers do not generate exactly the same signal when sequencing the same DNA content. So this means, given exactly the same 6-mer DNA content, and when we put it to the pore, or when we sequence it again as exactly the same in nanopore, the observation is that the signal readings are not going to be exactly the same. However, the another observation is that the signal readings are still going to be slightly similar to each other, because we're essentially sequencing the same DNA content, but they may be differing. The signal reading may be differing for several reasons. For example, due to the temperature reasons, or due to certain biological or the epigenetic reasons in the DNA molecule, such as, I don't know, certain molecule can be methylated. That's why we might be slightly reading the signal slightly differently, etc. So then the question is, or essentially, like, the DNA can be read like slightly faster or slower, depending on certain environmental situations at a particular time. So given these, how can we leverage this observation? So one idea is to do distance calculation, meaning after we generate these normalized values from the signals, what we can do is that we can concatenate all these normalized values to each other to create a certain vector, and then we could calculate the distance between vectors. And if the distance between two vectors are close to each other, then we can somehow infer that then these two readings are very close to each other, although they are not exactly matching, they are very close to each other. So perhaps maybe they are from the same DNA reading. But the issue is calculating the distance between vectors are essentially costly. So then the question is, can we do it more efficiently? So this is the main question that we ask to ourselves in the RawHash work. And hopefully the answer is yes, and this is how we do it. What we do is that we quantize the event values after we normalize them. So the goal is to include the similar values to the same buckets. Here the similar values are the normalized values that we generate, and the idea is those normalized values can be slightly different than each other, although they can be generated by reading the same DNA content. And what we want to do is that we want to assign the same value to these very slightly different normalized event values. So we call these values that we assign quantized values. So here are the steps that we perform to generate the quantized values. So assume that we have these two normalized values that we generated in the previous step. So you're seeing this particular value in its binary form, assuming the single precision floating point format in using the IEEE standard. So this is how it would look like in the binary form. And the observation is that if you remember the IEEE format, the first bit is the sign bit. It will tell us whether the value is positive or negative. The next few bits are the main part before the fraction, and after, I guess, like 8 or 9 bits, then we have the part for the fraction. Then the idea is that usually the most significant parts in this binary is more useful. It carries, let's say, the significant or most informative information to differentiate two values between each other. So by utilizing this information, what we do is that we take the most significant qubits. So in this case, we're taking the most significant 9 bits because the rest is just going to carry a little information, is the tiny fraction in these values that we really want to avoid, because we want to basically disregard those tiny fractions, because those tiny fractions or the tiny changes between two values can be caused due to the readings in the nanopore, although the DNA content can be the same. So that's why we take some more significant bits. And then the next observation is that when the values are too close to each other, then some bits, let's say, so I'm not going to give too much detail here, but the observation is that some bits that are in the middle are almost going to be exactly the same if basically the values are close to each other. So if they are not close to each other, of course, they can still be the same, but they are likely to be the same, and the rest of the bit information that we're generating could already be good enough for us to distinguish those such substantially different values than each other. So then basically, in order to further reduce the bit requirement that we're essentially generating from these 32 bits to a few bits, we want to basically essentially reduce them. So to reduce them, we apply this pruning step as well in the second part. Then basically the bits that we generate are called quantized bits. And essentially, although at start, these two 32-bit binary values were different than each other, because here that particular bit is 1 to tell us these values are actually then really different than each other, but they are essentially close to each other. So after the quantization steps, then the binary or the bits that we're generating from these 32 bits become the same by utilizing these observations that I just explained in these 3 steps. So then the idea is that then we can use these quantized values to identify whether two signal value are either exactly matching to each other or they are very close to each other. So we utilize this information in the next step when we're generating the hash value from nanopore signals. So our goal is to enable finding the efficient similar detection between signals by using these quantized values. So to achieve this, we pack the quantized values of consecutive k-mers or consecutive events that we generate from signal. And then, so this is basically the packed version of these quantized values of consecutive events. So you can assume that this is a 30-bit or 32-bit quantized value or 40-bit, it doesn't really matter, because then what we essentially do is that we use this packed quantized values with a hash function to generate the hash value of these packed quantized values. So what happens now is that if two different regions or two different signals are highly similar to each other, then the idea is that their consecutive events are going to be very similar to each other. Then this means that the quantized values that we're generating from these events hopefully are going to be exactly the same as well. Meaning, so we can pack them together to force them to exactly match all of these consecutive events so that we can use their corresponding hash values to identify these similarities between nanopore signals. So then let me cover the steps that we do in the evaluation, like how we evaluate RawHash. Maybe before that, I'll quickly check YouTube chat again, just to see if there are any questions. Okay, I'm not seeing any, so I'm going to move on. This slide covers our evaluation methodology. So we use data sets from very small, such as viral, to large genomes, such as human and metagenomics data sets. So we compared RawHash with Uncalled and SigMap. So all of these tools, including RawHash, do not require powerful computational resources to achieve efficient and portable genome analysis. So we have 3 use cases in our analysis. First one is read mapping. Second one is relative abundance estimation. Relative abundance estimation mainly tries to identify what is the percentage of a particular species or a particular genome in a given sample. And the third use case is contamination analysis. So the goal here is to identify whether the sample is contaminated by a certain genome. And we also show the benefits of the sequence until technique that I mentioned earlier, but didn't really describe what it is doing. So I'm going to also briefly describe in the next slides. So I'll first show the results related to performance. So here in this figure, we show the throughput results. Those throughput results are base per second. So here we show the throughput of a regular single nanopore sequencer, which can generate signal values up to 4,000. Recently, it's actually 5,000 signal values, which then can correspond to 450 bases per second. So then the goal is to actually be able to analyze at least that amount of bases in a second, so that we're always faster than the throughput of the nanopore sequencer. And if a certain tool for a particular data set is below that particular throughput value, then we can tell that safely that particular tool cannot perform real-time analysis for the particular system that we tested the tool on. So here we have these different data sets and use cases. We've tested using RawHash, Uncalled, and SigMap, and these numbers here show the throughput improvement compared to the throughput of the nanopore sequencer. So what we essentially observe from these results is that both RawHash and Uncalled can match the throughput of nanopore for all of the data sets. It's always faster than the throughput of the nanopore. So another observation that we make is SigMap falls behind the throughput of nanopores, specifically for large genomes. For example, the human genome, which is around 3 gigabases for the genome itself. So SigMap achieves 0.7x throughput of the nanopore device. And also, this is also true for the relative abundance estimation, which is a collection of the reference genomes that we evaluate here, which is then eventually essentially larger than the human reference genome. It was around 4 to 5 gigabases, if I remember correctly. So for all of these genomes, SigMap falls behind the throughput of nanopore, meaning it cannot perform real-time analysis for these particular genomes. And this is because it performs this costly calculation between the events to calculate the distance between the synthetic event values of a reference genome and the real raw signals, the real events that we generate from the raw signals. And it is essentially costly, and it's getting even more costly for larger genomes because there are many other more regions to check. And at certain point, due to these many regions, such a costly approach can fall behind the throughput of a nanopore sequencer. So given that, we then evaluate the benefits that we can enable using either RawHash or uncalled in terms of the sequencing time and cost. To evaluate this, we measure the number of bases that needs to be sequenced before making a decision to eject a read. So here, the lower is better, because this means that we needed to sequence only a fewer bases to make a decision, hopefully an accurate one, such that we spent less time and hopefully less money to sequence that particular read using a particular approach. So what we observe is that for smaller genomes, the uncalled is providing a better sequencing time and cost results. But for larger genomes, due to certain issues that the uncalled is essentially experiencing due to the many repeats or many regions that needs to be checked in larger genomes, it's basically essentially falling behind RawHash, and to make the accurate decisions that are assumed to be accurate, it essentially needs to sequence more, even more than RawHash. So for larger genomes, what we observe is that RawHash reduces the sequencing time and cost for large genomes than uncalled. So then let's quickly look at the accuracy results of genome analysis in these three use cases. Here in this part, we have the accuracy results for read mapping. Here we have the accuracy results for relative abundance estimation and the contamination analysis. And the accuracy results are in terms of precision recall and F1 score. The F1 score is essentially a summary of the precision and the recall values. So we mainly look at the F1 score to understand the main accuracy advantage of each tool. So here we have each tool here. So the main observation that we also make here, RawHash provides the best accuracy specifically for large genomes. For smaller genomes, the accuracy of RawHash is close to the other tools, but for large genomes, what we observe is that the accuracy of RawHash is significantly better than uncalled than SigMap. And this is essentially because it can make a particular raw signal unique enough, because it can concatenate many events to each other without reducing the accuracy, thanks to the quantization approach that doesn't specifically require the exact matches of signals, but it can also tolerate slight changes between event values, which is improving our sensitivity. So I'll quickly show the relative abundance estimation results as well in terms of the distance that we're measuring. So here we estimate the relative abundance estimation of each genome compared to the baseline as generated by Minimap2. And the distance that we calculate is essentially based on an Euclidean distance, or known as L2 norm, compared to the ground truth distance. So this is the estimated relative abundance ratios. So we provide a single sample including all of these species from COVID genome, E. coli yeast, and human genome, and green algae. And the ground truth is generated by Minimap2 here, and it is showing essentially the portion of each species included in the sample. So we use these estimations as the ground truth, and that's why the distance here is non-applicable, because we don't compare the distance to itself. Here we make also estimations using uncalled SigMap and RawHash. The estimations of the ratios of each species included in the mock community that we generated in our data set. And then after making the estimations, so these essentially create a vector of values. So we compare these vector of values to the vector of value that the ground truth generates, and then calculate the Euclidean distance between these two vectors. And what we observe is that RawHash provides the relative abundance estimation closest to the ground truth, which aligns well with our accuracy results that I showed in the previous slide. So the last thing that I'm going to go over is the sequence until mechanism. So I put it towards the end of the slides, because this is not essentially specific to RawHash, although it benefits a lot from the high accuracy and the high throughput of RawHash. However, the idea can essentially be applied to any real-time analysis mechanism. So that's why I want to create also this logic separation between the actual RawHash steps and the sequence until mechanism. So the key insight that we had is, like, we asked the question to ourselves, do we need to keep sequencing, sorry for the issue here, do we need to keep sequencing the entire sample for all applications in genome analysis, right? So the question is, there may be certain applications that we can make an accurate decision by partially sequencing the sample, meaning if we could stop the sequencing of a sample at a certain point, our analysis could still be accurate, and by stopping at a certain portion, we could also save from sequencing time and cost a lot. To give you an example, for example, can we predict the relative abundance estimation by sequencing only a portion of the sample and still provide accurate results? Potential benefits from such an approach, as I said, could be reduced sequencing time and cost by avoiding full sequencing. So we simulated such a benefit, and also simulated whether we can make accurate predictions by only sequencing a portion of the sample, accurate relative abundance estimations. And to simulate this, we randomly picked the mapping results using RawHash and uncalled. So these tools are generating certain mapping results for each read, and the entire mapping would correspond to the entire sample that we sequenced from a Nanopore sequencer. But if you had picked the randomly the mapped information, this could enable us to simulate what would happen basically if you had only sequenced those particular reads and then made the relative abundance estimation using these randomly picked mapping information from uncalled and RawHash. Here, the percentages show the portion or the percentage that we picked from the entire mapping results. Here, the entire mapping results would correspond to 100%, and here we are only picking 1 in every 4 mapping that these uncalled and RawHash is generating, and we're picking them randomly, to reiterate myself, because it's important to simulate the nature of also the sequencing. So here we reduce these numbers from 25% to 0.01%, and what we did is that we calculated the distance and essentially generated the relative abundance estimations, and then calculated their distance again to the ground truth. And the goal is to check whether the distance that we're generating using a portion of data is the same distance that we generated using the 100% of the sample, which we showed in our previous slide over here. So in this case, for example, uncalled, the distance of uncalled to the ground truth was around 0.1895, and RawHash was around 0.0847. So the question is, can we get close to these numbers so that we can make almost as accurate as estimations by sequencing the entire sample? And the answer that we find is almost always yes, depending on the portion of the samples that are being sequenced. So in the case of 25%, 10%, even with the 1% values, the distances that the estimations that we're making using these tools are pretty close to the distances that we generate using the entire the mapping information. Here 0.8 again, 0.18. Here up until the 1%, the values for RawHash is pretty close to 0.8, and for uncalled they are very close to 0.18. So then, based on this observation, how can we leverage this? So how can we decide that we should stop sequencing, for example, ideally after sequencing 1% of the sample, because we can very well generate almost as accurate results as accurate as sequencing the entire sample. And if basically the goal is to perform really cheap sequencing and to perform, let's say, relative abundance estimation that can tolerate such a difference between such a small distance difference between sequencing the entire sample and only 1% of the sample, then how can we basically decide at a certain percentage that we want to stop the entire sequencing? So that's where sequence until comes in. The sequence until is a mechanism that can dynamically analyze the results of a genome analysis use case to find the outliers in the analysis. Meaning, so at every point at a certain rate, we're always generating these relative abundance estimations using any tool, but in this case, we implemented sequence until in RawHash. And what we do is that after sequencing a certain amount of reads, we generate the relative abundance estimations, and then we try to identify whether the relative abundance estimation that we generated at a particular moment is an outlier compared to the previous estimations that we generated in the previous steps. So meaning we always keep a window of estimations that are generated in the previous time. Then when a new estimation comes in, what we do is that we compare the new estimation to the previous estimations to identify if there's an outlier. Meaning, if the estimation that we generated just caused a certain disruption in our analysis such that sequencing further reads are still providing some important changes to the estimations, such that we want to keep sequencing. But if there is no outlier in the previous estimations, then this essentially means that the further sequencing is unlikely to change the analysis significantly. Then, meaning we can hopefully stop the entire sequencing because we don't want to do further sequencing. So this essentially leads to significant reduction in sequencing time and cost. And we implemented this mechanism. We identified the outlier in real time for relative abundance estimation. And what we observe is that when we implement sequence until with RawHash, we can stop the entire sequencing after sequencing 7% of the sample. So this decision is made in real time, dynamically, because in reality, we don't really know what percentage we sequenced so far, right? So we can only tell whether the further sequencing is sufficient or not. So we just stop the sequencing when deciding the further sequencing is not necessary. And then we check what percentage this is corresponding to, like the number of reads that we sequenced so far, what percentage they are corresponding to the entire sample. And what we observe is that this is the decision that we're making is around 7%. We stop the sequencing after sequencing 7% of the reads. And what we observe is that the estimation that we're making is extremely close to the estimation that we would be making if we had sequenced the entire sample. This distance is showing the distance using sequence until technique to the estimations to the estimation basically if you didn't use the until technique. Then what we observe is that the sequence until dynamically stops the entire sequencing after sequencing only 7% of the entire sample while providing high accuracy. Sequencing only a portion of the sample significantly reduces the sequencing time and cost. So here you can assume that if we had sequenced the 7%, then this would lead us to around 15x reduction in sequencing cost. So with that, I'll conclude. So let me reiterate the summary or the executive summary of RawHash again. So the problem in nanopore sequencing is to perform real-time genome analysis, and they are usually inaccurate or inefficient for large genomes, causing serious barriers and fully exploiting the opportunities in real-time genome analysis. So our goal is to enable efficient and accurate similarity identification between raw signals. RawHash encodes the similar signal values into the same quantized value to elevate the noise issues in raw signals. It then generates the hash values from quantized values to efficiently identify similarities between signals based on the hash value matches. In RawHash work, we also propose a technique called sequence until that can accurately and dynamically stop the entire sequencing. RawHash performs high throughput analysis. It generates the most accurate and efficient analysis for large genomes, and it can reduce the sequencing time and cost by 15x. So the RawHash is accepted to appear in the proceedings of ISMB ECCB this year. So you can again find the QR code to the preprint version of this paper. Here we have the source code of RawHash as well. We provide all the scripts to fully reproduce and easily reproduce all the results that we show in our paper. With that, I'll conclude RawHash. So before giving the floor to Banu for the TargetCall presentation, I'll check whether there are any questions in the YouTube chat. I'm not seeing any questions. I'll wait a few more seconds. Maybe I can stop sharing in the meantime so that Banu can get ready for her presentation.
Hi, John, can you hear me?
Yes, I can hear you.
Okay. Okay, do you see my screen in the presentation mode as well?
Yes, I do your screen in the presentation mode. Everything looks fine for me. And I don't see any questions also on the YouTube chat.
Should I
so
start?
You can start anytime you want, basically.
Oh, but the slides are not in the presentation mode. Okay, they are not. See.
Oh, yeah, on my end, yeah, they are in the presentation mode.
Okay, I was just checking YouTube, it's a bit delayed. So maybe I need to quickly start. Hi, I'm Banu and I will present TargetCall, which is analyzing these nanopore signals in a different way. The title of our work is Eliminating the Wasted Computation Basecalling via Pre-Basecalling Filtering. And our summary is, so we realize that basecalling can consume up to 84.2% of the total execution time in entire genome analysis pipeline, and it becomes a bottleneck for this pipeline. But what's the real problem is that majority of this costly computation is wasted, as majority of the reads do not match the reference of interest by the application, and they are wasting the basecalling computation. So what we aim in this work is to have a method that would eliminate this wasted computation basecalling while maintaining high accuracy, scalability, and adaptability. Our idea is to have a pre-basecalling filter that would filter out the user's read before basecalling with a highly accurate filter, with high accuracy and high performance. For this, we propose TargetCall, which is a new pre-basecalling filter that works in 2 steps. First, we have a lightweight basecaller, which we call as LightCall, that performs basecalling in a noisy way compared to state-of-the-art basecallers. And then second, there's a similarity check component that compares these noisy reads to the reference genome to determine the similarity of these reads and kind of find out if the read is useless or useful. Our results show that with TargetCall, we can improve the end-to-end performance of basecalling by 3.3x compared to state-of-the-art basecallers by filtering out majority of the useless reads. And we show that if you were to repurpose the adaptive sampling methods that John presented or discussed as pre-basecalling filters, TargetCall achieves better performance, throughput, recall, and precision than them. And we open source our work in our GitHub page. I will start with background, but John already gave a lot of the background, so I will skip these parts. As John said, genome sequencing is important in many areas, and nanopore sequencing is a widely used sequencing technology that has long reads, high throughput, and low cost. A traditional nanopore sequencing pipeline starts with genome sequencing, where the reads are passing through a nanopore, and as this is happening, nanopore sequencers are generating a raw signal. This raw signal then basecalled using complex neural network models that would convert these raw signals to a set of bases. And the next step after this is to perform read mapping, where these reads are matched to the reference genome to compute if the read can be mapped or not mapped to the reference genome. And prior studies shows that only the base calling step can consume up to 84.2% of the total execution time, while the remaining analysis takes less than 16%. That's why we conclude that base calling is really a major computational overhead in nanopore sequence analysis pipeline. But our real observation in this work is that majority of these base call reads are discarded after base calling in the later steps of the downstream analysis. So one example for this applications is the SARS-CoV-2 genome assembly, where only less than 1% of the reads are useful, while the entire read set is processed. And typically the reason why 99% of the reads are used is that they do not match the reference genome that is targeted by the application, which is SARS-CoV-2 reference genome in this use case. That's why we say that users reads waste significant amount of base calling computation in traditional pipeline. One option is targeted sequencing or adaptive sampling, as John presented, which is in contrast to the traditional pipeline where we sequence all reads and base call all of them. In targeted sequencing, what we do is we selectively sequence only the useful reads. And for this, we add a step in the sequencing part where we identify if the read is going to be useful, and then if it's not, the nanopore sequencers can stop the sequencing of the user's reads by rejecting them from the pore. But this requires a method, and as you can see, the rejected ones are stopped and the accepted ones are fully sequenced and then sent to the base calling step. But this requires a method to identify which reads are useful and which reads are going to be uses. And some of these methods are presented by John, such as OnCall, SigMap, and RawHash. But we identify several limitations of these prior tools, and we state that all of these approaches at least suffer from one of the three following key shortcomings. First, they have low sensitivity and they falsely reject many useful reads, or they have poor scalability in the sense that their performance and accuracy drop significantly for large reference genomes, or they have lack of adaptability, which means that for each kind of genome sequencing application that you are targeting, or each kind of different reference genomes that you're targeting, you need to retrain neural networks, which is a costly method to do. That's why we say that these targeted sequencing methods, although they are useful for real-time analysis, they are not robust approaches for eliminating the wasted computation in base calling. What we have as our goal in this work is to eliminate the wasted computation in base calling, but while maintaining high sensitivity in keeping useful reads, and while being scalable to large reference genome, and while maintaining adaptability to different use cases without requiring any further preprocessing steps. Next, we present TargetCall, which is our pre-basecalling filter. If you remember our approaches, our first option was to have the traditional pipeline where we sequenced and base call all reads. And our second option was to do targeted sequencing, where we base call everything that is sequenced, but we selected the sequence things depending on if they're going to be useful. There's a prediction step. And then we only do sequence the useful ones, or the ones that are predicted as useful, and then we base call everything that's coming from the sequencing step. What we propose instead is to have a pre-base calling filter, which is to selectively base call the useful reads. So in contrast to the traditional pipeline, we sequence everything similar to the traditional pipeline, but we don't base call everything. And in contrast to the targeted sequencing, we sequence everything, but the targeted sequencing does not sequence everything. But the thing is, this requires a method or another step in the pipeline, the pre-base calling filter between the sequencing and base calling step. And this filter needs to identify whether the read is going to be useful or useless with high accuracy. And in TargetCall, anything that is identified as users are discarded from the further analysis in the pipeline, and useful reads are forwarded to base calling step to maintain the base calling accuracy for the useful reads. So in high-level overview, TargetCall works in two steps, and then with a high-level goal of predicting if the read is going to be useful. And the first step of TargetCall is LightCall, which is a lightweight base caller, and it does base calling, but in a noisy way. Noisy in the sense that the accuracy of individual bases are not as high as the state-of-the-art base calling, but it's still high enough to compute if the read is going to be similar the reference genome if you were to do the state-of-the-art base callers. And after light call computes the noise reads, there's a second step, which is the similarity check component that compares these noise reads to the reference genome to predict if the read is going to be useful. And as I said before, anything that is useless is discarded from further analysis in TargetCall. Anything that is useful is forwarded to the base caller so that we maintain the base calling accuracy for the useful reads. In contrast to the prior methods, TargetCall has high sensitivity because we have a highly accurate similarity check component. It has good scalability because the light calls model's performance is independent of the reference genome size, and it's adaptable to different use cases without requiring any other processing step or any other neural network training. Because the light call model is generic, that means like you just use the same light call model regardless of your read set and the reference that you are using. And that's why TargetCall is a robust solution for eliminating the wasted computation and base calling. Now we can explain our two steps in a bit more detail. And the first step is light call, which is a lightweight base caller that produces noise reads compared to the state-of-the-art base callers. And light call is designed based on simplifying state-of-the-art base caller model architectures while maintaining most of the accuracy benefits, but reducing the execution time significantly. And we made this simplification in 3 steps mainly. First, we reduce the channel size of the convolution layers. Second, we remove the skip connections. And third, we reduce the number of basic convolutional blocks in the state-of-the-art neural network models for base calling. And the final light call architecture is composed of repeated convolutional blocks, where each block has 1 or 2 components that include group convolution, pointwise convolution, batch normalization, and read layers. And the second component is similarity check, which has input of a noise read coming from light call and the reference genome that is determined based on the application that we are targeting. And the goal is to find if these two things are similar. And depending on the answer, the read is going to be useful or useless. So in some applications, the read is going to be useful if it's similar, and in some other applications, the read is going to be useful if it's not similar to the reference genome. But for this model, we didn't design anything new. We just used the state-of-the-art read mapper, Minimap2. And we showed that in the paper while using light call and similarity check together. So similarity check or Minimap2 using the noise reads coming from light call, we can achieve up to 99.45% sensitivity in keeping useful reads. So that shows that like the minor or the noise reads coming from light call is not noisy enough to hinder our ability to find if the read is going to be useful. And we conclude that and show that in the paper that 0.55% of the loss of the useful reads during the filtering time can be tolerated by sequencing depth of coverage that is already required for accurate genome sequence analysis. And we evaluate TargetCall using 3 different use cases. We choose 3 different use cases in a way that we show how scalable and adaptable TargetCall is. Our first use case is SARS-CoV-2 detection, where we have a small COVID reference genome. And our sample of reads contains both COVID reads and human reads. And our goal is to identify COVID reads among this sample. So we want to eliminate the human ones. And if the read is now in this use case similar to the reference genome, which is the COVID reference genome, the read is identified as useful because we are targeting COVID references, and otherwise the read is identified as useless. Viral detection is also a similar use case, but this time the difference is our reference genome is not one single viral reference genome, but it's composed of multiple of them. So we have a bit more complex reference genome, and our sample now contains bacterial and viral reads, and we want to identify the viral ones. Here now the viral and bacterial reads are resembling each other more compared to human ones. That's why it's a bit like a different use case. But the approach is similar, and anything that is similar to the reference genome is identified as useful for this use case as well. What's different is the sepsis detection use case, where we have a large human reference genome. And this is to show that like TargetCall can scale well to the large reference genomes, and we have a human reference. And what we want to identify is all the bacterial reads in a sample that has bacterial and human reads. The difference is this time we don't know which bacterial reads we are looking for. Basically, we are checking for any kind of bacterial read in our sample. That's why instead of searching for a specific ones, we just filter out what we don't want, which is the human reads. And that's why in this use case, anything that is resembling the reference genome is identified as useless, unlike the other ones. For our evaluations, we have two sets of experiments. First, to show the benefits of pre-Basecalling filtering, where we have the baseline as Bonito, which is a state-of-the-art base caller, and we compare the Bonito alone and Bonito preceded by TargetCall to perform pre-Basecalling filtering. And our evaluation metric is Basecalling speed up, at least for this presentation, and you can find a bit more results in the paper. And the second set of experiment is to compare TargetCall against targeted sequencing approaches. And we also compared it with OnCall and SigMap because these are the ones that are fully open source and also can be applicable as pre-Basecalling filters without any other changes. And our methodology is to repurpose these methods so these methods also require a labeling mechanism to predict if the read is going to be useful and then rejecting the useless reads. We just repurpose these labeling mechanisms to perform pre-Basecalling filtering instead of adaptive sampling. Basically, we just compare TargetCall, Bonito pressed by TargetCall, OnCall, and SigMap in terms of their execution time, recall, and precision, and more in the paper for throughput. We have various data sets, 5 different resets from various organisms, most of them sampled from prior work, and then one we simulated using Deep Simulator because we couldn't find any proper real data set for this type of viral reference mix. And we open source all our data sets so that you can reproduce our results. We have 4 different reference genomes with various reference genome sizes and ratio of useful reads to show that we really can do very well in different kind of scenarios. So we evaluated TargetCall using 2 GPUs, NVIDIA 100 and the Titan V for light call, an AMD system with 0.2 terabyte main memory for the similarity check component. But for evaluating SigMap and OnCall, we use a similar AMD system with 1 terabyte main memory because SigMap and OnCall require more memory than we have in the first system for large reference genomes. Our first metric is the Basecalling speed up that we can achieve with TargetCall. So as you can see in the y-axis, we have the execution time, and in the x-axis, we have different use cases as well as the average. So this blue bar shows the execution time of Bonito for each use case and the average. And then the green bar shows the TargetCall execution time plus the Bonito execution time for reads that pass the TargetCall filter. So it's really an end-to-end comparison of the Basecalling times, and we conclude that on average we can get a 3.3x speed up, Basecalling speed up over Bonito, and show that TargetCall is really beneficial for improving the Basecalling execution time. And when we compare TargetCall against state-of-the-art approaches in terms of its performance, again, all bars include both the execution time of each specific tool and the Basecalling time of the reads that pass by these individual filters. Our first conclusion is that TargetCall can provide up to 9.7x speed up over prior targeted sequencing approaches. And more specifically, the speed up improvement is speed up improvement is larger for larger reference genomes, more specifically up to 13.3x over SigMap, and we were even unable to compare it against OnCall because OnCall couldn't generate the index for such a large reference genomes. And next, we compared the recall of these tools, and recall is referring to the ratio of keeping useful reads among all the useful reads. And on average, TargetCall's recall is 99.1%, which is quite high. And it's actually quite better than prior approaches, up to 23.2% better recall over SigMap. And similar to the performance benefits of TargetCall, recall benefits of TargetCall also improve with increasing reference genome size, where we can provide up to 15% better recall compared to prior tools on average. So there's one caveat in this plot, which is that in sepsis use case where we have a large reference genome, we don't actually measure the recall for finding human reads because here the useful reads are the bacterial reads. So if if the human reads is not matched to the human reference genome by this use case, they just pass the filter in a wrong way, and it's not a problem for the bacterial reads that are the real useful reads. But what we wanted to test is what if we had a use case where the goal is to keep the human reads in a sample, and we have a human reference genome as well, and then we find that like the recall for finding human reads for sigma and uncalled is quite low. It's like on average is like 47% or something. And this is recall for finding human reads in a human sample, whereas TargetCall provided more than 96% recall for the same scenario. That's why we conclude that TargetCall consistently provides high recall for all reference genome sizes tested, regardless of the type of experiment that we designed. And lastly, we compare the precision of these TargetCall against these adaptive sampling approaches. First, we concluded TargetCall's precision is on average more than 92%. Which shows that TargetCall can really eliminate the wasted computation in base calling by filtering out the user's reads before the base calling step. And on average, our second conclusion is that on average, TargetCall can provide up to 60% better precision than prior tools. We have more details in the paper about like details of how these targeted sequencing methods works individually, and then what are their limitations, and what is our light call design in a more specific way, and why we chose to do a specific type of optimizations while improving the performance of the light call, and then details on our evaluation methodology, and more detailed results on different target call designs, many different light call architectures that we used for target call design to see the base calling speed up, recall, and precision of these different light call architectures to finalize our design, and end-to-end accuracy and performance improvement of target call on entire genome analysis pipeline, including the variant calling step, and throughput comparison of target call against our sigma and uncall, where we show sometimes more than 1100x better throughput than prior tools, which really shows that target call can be applied for real-time analysis as well. And a peak memory discussion between these tools and then target call, which shows that target calls like peak memory requirement is much less than prior tools. Please check out the paper for more details about our results and everything else that we discussed. And check our GitHub page if you would like to test target call and verify our results or reproduce our results. And let me conclude with that. So target call is an accurate, scalable, and adaptable pre-based calling filter that has two-step approach. First, a lightweight base caller light call that does noisy base calling with high performance, and second, similarity check component to compute if the read or predict if the read is going to be useful for the later downstream analysis by computing the similarity of the noisy read coming from light call to the reference genome that is identified based on the use case. And our evaluations of target call shows that we can improve the base calling performance significantly for all use cases that we tested by filtering out majority of the user's reads. And we also show that target call has better recall, precision, performance, and throughput than these adaptive sampling approaches if you were to repurpose them as pre-based calling filters. And I think with that, I'm done. So I think there is no questions in here or YouTube chat. I'm going to wait for 10 seconds or so, and then I will stop the session if there is no question in YouTube. Okay. Thanks everyone for attending. And I think with that, we conclude our semester. Have a nice summer and